Our partners in joining, I already see a lot of you, a lot of familiar people, a lot of familiar partners in the list of people who join. So I see the partners from Bulgaria, Egypt, Morocco, Germany, and Austria, of course, Ukraine. I think Michelle from Cyprus and partners from Greece. So I'm very happy that we have so different and interesting audience today. And I think Jan from probably from Kenya. So let's slowly start our today's webinar. So the topic of today is inoculation. First of all, we speak about legume crops and soybeans. It's not something unknown, like in some countries, as we will see today, the rate of inoculation reaches up to 80% of all soybean and legume crops. But still, there are countries and regions where there is still space for development, even in Ukraine. Not all farmers use inoculants still. So we would like to share today with you our experience and our knowledge about this topic. So give me a second, I show you the screen. Yeah. So what we will speak today about, we will speak about in general the history of inoculation and industry trends in this area. We will speak about some general industry trends and figures, like how much soybeans are in the world and is it a growing market or not. And then we will speak about different technological aspects of how to improve efficacy of your inoculants application, what could be average increases of the yield in different technologies from our experience, what could be approaches to improve efficacy of inoculation using different protectants or synergists to the inoculants. And then also we will share with you our existing experience in different crops and regions. So for those who see us first time, I don't think there are a lot of such companies, but still, let me remind who are we. So we are BTU, a group of companies with origin in Ukraine, but also with office in Germany. We have more than 500 employees. We use inoculants on not only inoculants, but biologicals on more than 4 million hectares each year. In general, we work not only with inoculants, but with more than 60 products. And we have a big database of trials with more than 3,000 of them with quite high level of success rate. So as I mentioned, we also have office in Germany. In general, we are present in 25 countries through our partners, and mainly in Central and East Europe, but we are also developing in North Africa, Latin America, and some Asian countries. We have around 25 biological registered in European Union, and of course, those 25 includes inoculants. This is how our factory looks like in west part of Ukraine. We have quite huge production capacities, so we have space to supply to new markets. Quality is very important for us, and so we have special equipment and technologies to provide stable, sterile, and clear and effective solutions based on microbes. This is our office in Kyiv and part of our team and our international team and office in Germany. I'm glad that this year we made a meeting of international partners here, and a lot of you already visited our location in Germany. We are quite active in the industry on events like World Biostimulants Congress, BioAG events, ABIM or DLG, etc. So you can find us in a lot of these locations, and also we try to gather experience from all around the world and use most novel approach to share with our partners. Again, on international market, we rely on our partners, distributors, on their knowledge, on their teams, on their background and market understanding. That is why we look for strategic partners with whom together we introduce biological solutions in this or that area. So we start from identification, registration, tests, patenting, experience exchange, trials. We help also to monitor them and to understand, to prove that product work in local conditions. We have a separate marketing team and a lot of marketing materials which we share with you. And we gather our partners together once per year to exchange our international experience in biological application. So let's come closer to today's topic about inoculants. So speaking about inoculants, most of all we speak about legume crops and among them like no different legume crops, but also soybeans, which are the plants which normally are inoculated. So in general, this is quite a huge market of more than 260 million hectares spread around the world. But what is interesting to show or pay attention to that this market is growing. If we compare like the areas where the soybeans were grown in around 2000 years, and now it's almost a double growth. And if we take main legume crops on this, which you can see in the table, they also grow, not such significantly, but still quite a lot. So there are several like factors which influence this. One of the first factors is the growth of population and the importance of nutritional value of legume crops. But also legume crops are quite good for soil properties improvement because these crops normally fixate nitrogen by their roots and enrich soil with nitrogen. That is why even in extensive and intensive technologies in Europe or around the world, after, for example, sunflower or corn or wheat, soybeans are quite popular in rotation. Speaking about the market of soybeans, also it's important to understand the factors which influence the growth of these crops. So one of the factors, of course, is the price of this crop in comparison to like other crops and historical value, because as we see now is more or less quite low rate of soybean prices. It's much lower than it was in like around 2022. And quite often also prices and tendencies on the legume crops and soybean markets are related to the price of nitrogen and nitrogen fertilizers. Because when farmers grow soybeans or legume crops, they don't need a big amount of nitrogen because again these crops fixate nitrogen by themselves as well. And here we do some interesting calculations, which you can see here. So if you apply nitrogen, like for example, urea or some other nitrogen fertilizers, the price of a kilogram of nitrogen could be like around $1 per kilogram. But if we recalculate nitrogen, which you can get from soybean and legume crops as a result of seeds inoculation, the nitrogen which you get is like much cheaper. So that is an economic way to get additional nitrogen to your soil and to your plants by using nitrogen fixing bacteria in comparison to conventional nitrogen. Also, if we speak about inoculants market, it's difficult to find some specific figures. There are different industrial experts, for example, Dunham Trimmer, which we rely to a lot. So in 2021, they evaluate inoculants markets to be around $500 million per year. And with projections of the growth of this category, we estimate this market to be now around 700, $800 million in the world. Also, it's important to understand how this market spread. And speaking about soybeans, of course, a lot of you know that the biggest market is in South America, where there are millions of hectares of soybeans in Brazil, in Argentina, and other countries. Also, this market is followed by Canada and USA, with quite huge areas and territories under the soybeans. But also Asia is quite a big market too, followed by Europe and Africa. What is interesting to mention that speaking about pulses, situation is a little bit different, and it's a little bit even surprising for us that the biggest market for growing of pulses is Asia and Africa, and followed by America and Europe. So again, a lot of our partners in Europe, etc. So actually, we are quite small in terms of legume crops and soybeans growth. That is why, for example, of course, Ukraine is quite a big grower with around 1 million hectares, but in all European Union, there are just a few million hectares of soybeans, which is not so huge comparing to other parts of the world. But again, it's interesting to see that Africa is a growing market, and I hear a lot of projects like in West Africa, in North Africa, to really invest in huge new territories to be where pulses or soybeans are going to be planted. And I'm very glad that today with us are the partners from these areas as well. So on this, I would like to give a word to my colleague, Yaroslava. Probably you remember her from our previous webinars and event. And Yaroslava will tell us more about inoculation and everything about that.
Hello everyone, thanks for introduction. Do you hear me well?
Yes, we hear you well.
Perfect. So, yes, today I'm going to tell you some more facts about inoculation and how it's going on, about this amazing symbiosis between plants and microbes. So I hope we will get something really useful for you today, and afterwards my colleagues will continue with some practical cases and we will see how these microbes are working in fields. So let's start with basics. What are rhizobia? So actually, rhizobia is not a single species of bacteria. It's rather a group of sometimes even unrelated bacteria, which are not, which are not closely related, which are known to be able to fix nitrogen from the air. They have developed this ability because they were evolutionarily adapted for different environments with low excess of nitrogen, and they had to develop this actually very complex and resource-consuming process to provide themselves with nitrogen. And you also might know that there are free-living nitrogen-fixing bacteria like Azotobacter, and rhizobia are bacteria which form symbiosis with plants, with their roots specifically, to create a favorable environment for nitrogen fixing. And it's very important to note that rhizobia cannot fix nitrogen independently. They always need a host plant. You can see some pictures of how nodules work and how biologists are staining these nodules to see how bacteria are living inside of them. And obviously this symbiosis is a win-win cooperation. while plants are getting nitrogen from these nodules, from nitrogen-fixing bacteria. These bacteria on their side are getting organic compounds, especially organic acids from plants as a food source, and that's how they work together. This symbiosis is known to be very old. It's extremely old, and actually, this symbiosis has been used by humans for thousands of years. Our ancestors didn't even know about these microbes, but what were they doing? They were transferring some soil from fields where legume crops were growing because they noticed that after legumes, other crops are giving better crops, better yields. And they made a conclusion that legume crops are doing something positive to the soil, and if the soil is transferred to other fields, it transfers better its better characteristics, and that's why they were actually first first agricultural biotechnologists many thousand years ago. Obviously, now we are doing it in a more efficient way, but the biological base is pretty similar. So, what else do we have to know about rhizobia? Is that the productivity of nitrogen fixing in different crops is different. It depends on plant characteristics, on above ground biomass. As you can see, alfalfa is a champion in this rank. And another interesting fact that average increase of soil nitrogen per hectare might reach up to 700 kilos, 70 kilos, excuse me, 70 kilos when we are growing pulses, like soybean, for example. And scientists have also estimated that each year, all around the globe, about 40 million tons of nitrogen are fixed by legumes in symbiosis with rhizobia. That's an amazing amount, I would say, and actually I cannot overestimate the importance of this process to the life on Earth, because this is the way how we're getting amino acids, and I will show you later how. So let's dive a little bit deeper into biological mechanism of how this symbiosis is actually formed. On the right side, you can see a pretty simple illustration how this process is going on. So we have legume plants, we have some bacteria inside its nodules, and we see this exchange that bacteria are giving nitrogen compounds to plants. Plants need nitrogen to grow and to form their proteins, and plants are given organic acids to bacteria. But how the symbiosis is formed? So in a soil, roots of legume plants, they secrete flavonoids. Flavonoids are very specific to each species of legumes, and when near the roots there are some rhizobium bacteria species, they can sense these flavonoids, they can feel them. And when they feel these flavonoids, they know that legume plant is growing nearby. And what happens next? There are some specific genes in rhizobium bacteria, which are called nod factors, not genes. When rhizobia is activated by flavonoids, these genes start working. They are on their side secreting nod factors, so they're basically communicating. So plant is communicating to bacteria using flavonoids, and bacteria is communicating with plants using nod factors. And they know, they are telling each other they are close, and after that they interact with plant root hairs, and this process actually is pretty similar to kind of infection. So rhizobia have to slightly be tricky with plant immunity not to be killed by plant. They communicate a lot about it, and afterwards this infection thread is formed, and root cortex cells are involved to create a nodule. And this biological process, this is a scheme of reaction. As you can see, atmospheric nitrogen and a lot of energy in form of ATP is used to create ammonia, and mostly ammonia, which is most interesting for all of us. And that's how it works in nature. Let's talk a bit more about nodules and how do they do their job. Basically, there are two types of nodules. Some of them can grow undeterminately, so they don't stop their growth during their life. Some are determinate, so some nodules are round-shaped. It means they reach some specific size and afterwards they stop. Here you can see indeterminate nodules, and basically the same process but shown here in slightly different way. So we have roots, we have rhizobia attaching to roots, forming infection thread, and after that, when bacteria penetrate plant root cells, they change into bacteroids. It's a special form of root cells, which creates a favorable environment for rhizobia. Very important moment is that process of nitrogen fixing requires anaerobic conditions, so no oxygen should be present. And it's a problem. Why? Because we have oxygen in the soil, we have oxygen in plant tissues because plants are aerobic. How this dilemma was solved? In bacteroids, bacteria and plants create anoxygenic environment. They do it with the help of hemoglobin-like protein called legume. And this protein works, it does the same job as hemoglobin does in our blood. It binds oxygen. But in our blood, hemoglobin is binding oxygen to deliver it to our tissues and cells and so on. While in plants, hemoglobin is used to bind oxygen and to fix it, to keep it, and to allow nitrogenase, a key enzyme for nitrogen fixing, to work. So when you see pink or reddish nodules, it means hemoglobin is working and nitrogen fixing is also working. And when you see it's green or white, it means there is no nitrogen fixing process in this nodule. So I told you a lot about nodules and symbiosis. And as for many symbiosis, this relationship is pretty specific. I mean, different species of rhizobia are specific to different species of legumes, like soybean, common bean, pea, lentil, so on and so forth. So yes, there are some species of rhizobia which can form symbiosis with few species of legumes, but typically there are examples of the most efficient symbiosis between these two, because as you can remember from previous slides, they have to precisely exchange with their chemical and biological signals to form the symbiosis. That's why unrelated species will not form symbiosis, because they will not understand each other. For example, soybean, the most popular bacteria for soybean inoculants is Rhizobium japonicum, which is the most widely used all over the world. And for others, another very popular species is Rhizobium leguminosarum. And this bacteria has at least 3 biovars. What is biovars? It's kind of like strain, but a broader term. It's a set of strains which are specific to different plants. For example, Trifoli is specific to clover, Faseoli is specific to common bean, and so on and so forth. So they have to be really specific, otherwise they will not work. I have already mentioned nitrogenase. Nitrogenase is key to successful nitrogen fixation. It's a huge enzyme. Here you can see only small region of this enzyme. And what I wanted to show you here is its heart. It's a functional center that runs the entire process of nitrogen fixation. And you can see molybdenum here. This metal is crucial for nitrogenase activity. On the right side, you can see a diagram how nitrogen fixation rate depends on this metal. You can see two other metals like vanadium and iron. Yes, in some conditions, when molybdenum is not present in the environment, plants will substitute it with vanadium or iron, but the efficacy of this enzyme will drop significantly because they need molybdenum specifically. That's why molybdenum is very important for legume cultivation, and don't forget about it. It's very important for nitrogen fixation. And just a fun fact, some biological fun facts. Find some differences. Here are protein structures of hemoglobin, which we have in our blood. It's myoglobin that we have in our muscles. And this is black hemoglobin, which is in legume nodules. You can see they're pretty similar and they're binding oxygen, these two balls. Yeah, Mother Nature is incredible and impressive. And as we are already talking about living creatures, all living creatures need some optimal conditions for their life, and rhizobia are even more picky than other bacteria because rhizobia do not form spores, like for example bacillus. And it means they are pretty fragile, and all these conditions should be followed for them to survive. So firstly, how to uh, provide successful inoculation. Firstly, you have to choose a specific species which is relevant for your crop. At BTU, we have a huge museum, huge bank of strains, and we select species for each type of crop to ensure successful inoculation. Secondly, it's very important to follow manufacturer recommendation. I mean, regarding storage and transportation of inoculants, because again, they're not forming spores. They're pretty fragile. So temperature regime and other conditions should be obeyed. Third one is very important to use protector when possible, because protectors are specific compounds which help rhizobia to survive on seeds, especially now when almost everyone is using seed chemical treatment. It's very important to follow all these conditions to make rhizobia live successfully on your seeds until they are put into the ground. And also, you have to avoid exposure to direct sunlight because they cannot survive it. And the last but not least important is to get your inoculants from reliable supplier who is responsible for what they're producing and how they're storing and how they're transporting it, so you get living and effective bacteria. Let's talk also a bit more about other factors of environment that affect rhizobia. And soil pH and environment pH is one of the most evident factors, most easier to measure. So as you can see from this heat map, there are some species which are more resistant to acidic pH, while some of them are less resistant. The optimal pH for most rhizobial inoculants is between 6 and 7, and even it may be slightly alkaline, while strongly acidic soils inhibit this process of symbiosis. But I would say that acidic soils are also bad for legume plants, not only for rhizobia. So you have to consider it when you choose a field for planting them, I mean legumes. And you can see on these graphs a correlation between soil pH and number of, on this graph, of P. rhizobia. So we can see the best results we get at neutral soil reaction, and same for of alpha in acidic soils. A number of plants which have nodules is 0. So this symbiosis is not working, and I bet plants are also not doing well at this pH. And let's now a little bit broaden our biological topic. So not only rhizobia live in the soil. We know soil is rich with numerous microbes. There are millions of species of different microbes in the soil. There are mycorrhizal fungi. There are many different bacteria, actinomycetes, and so on and so forth. And all of them, they interact in the soil in nature. And during past recent years, a lot of research has been done on this topic, on how we can even increase the efficiency of inoculation. And now co-inoculation is a new trend in agriculture. So basically, what is in co-inoculation? It's when you treat seeds not only with one species of nodule-forming bacteria, like Bordetella japonicum in case of soybean, but you also inoculate these seeds with other plant growth promoting bacteria or plant growth promoting fungi. So when we create this microbial consortium, it increases the efficiency of symbiosis, and it also increases the productivity of plant, and it's resistant to environmental stresses. So it's obviously very beneficial practice. And this practice is being widely adopted already in the world. So here are some slides from last year presentation at Biostimulants World Congress by Jose Roberto Pereira from Brazil. He told us about co-inoculation of soybean in Brazil using Bordetella and Azospirillum. So you all know how many hectares, millions of hectares of legumes are in Brazil. And you can see some statistics that in Brazil today, 88% of their fields are inoculated with rhizobia inoculants each year. You might have heard that when you plant soybean after soybean on some other legumes after legumes, you might think that there are already many rhizobia and you don't have to inoculate, but the scientific research tells us it's not enough, and you have to inoculate seeds each year with most efficient strains to have the best results. And you can see statistics in Brazil. They did a lot of trials, and now most of their farmers are inoculated no matter how many years of legume cultivation has this field in their history. And look at lower graph, it's about adoption of co-inoculation. You can see its rate is growing at pretty high speed. So last year they already had almost half of their areas inoculated. They were already using co-inoculation. And according to speakers' opinion, this trend will be growing because farmers see positive results, and this recommendation to co-inoculate soybean is also promoted by their soybean growing organization. and it's actually providing really beneficial results. So how it works, the mechanism is pretty logical. So when you inoculate one plant with few different beneficial organisms, uh, they uh, they produce more biologically active compounds. They stimulate plant stress tolerance. They improve plant development and growth. They stimulate bigger biomass accumulation, and that's why plants have a better resistant to unfavorable environmental conditions, and they have bigger yields. And you can see also the slide from Biostimulants World Congress. It's a comparison of soybean plants which were inoculated. This one was inoculated only with Bradyrhizobium, and these plants were co-inoculated with combination of Bradyrhizobium and Azospirillum. And look at how their root system is developing, how many additional roots are formed, and that the main root is going deeper than in just mono-inoculated plants. And I think now it's evident why they're using it. And look at year 2019, they are doing this research for 7 years already. And now let me share you some examples of which products from BTU can be used together with our inoculants. I will start with mycorrhizal fungi, with mycofriends, those who um, have attended our previous events about mycorrhiza can remember this photo of mycorrhized roots with arbuscules and vesicles formed by mycorrhizal fungus. And when you inoculate your legume crop with rhizobia inoculant together with mycorrhizal fungi, you have a synergistic symbiosis. So plants have better root system development due to mycorrhiza. They have nitrogen fixation due to rhizobia, and altogether they improve immune status of plant, and plants increase the efficiency of nutrient consumption and water balance. So this is a win-win-win symbiosis. Another example of co-inoculation is using humic acids, humics and fulvic acids. We all know that these compounds are crucial for soil fertility because they can carry nutrients in the soil and they improve soil qualities. And when they are used together with inoculants for legumes, humates, they create a favorable environment for rhizobia bacteria to survive and to colonize the roots. It actually increases the efficiency of inoculation, and it also improves nutrient uptake, and nodules are formed faster, they're more active, and this is how it works. And another option for co-inoculation is co-inoculating soybean or other legume crops with rhizobia together with Bacillus thujalensis. What this type of co-inoculation does is when Bacillus thujalensis colonizes roots in the soil, it creates a protective barrier against pathogens which live in the soil, and in this way they protect plant roots. And while Bacillus thujalensis protects and stimulates root growth, Bradyrhizobium japonicum or other rhizobia are doing their job, inoculating roots and doing nitrogen fixation and providing plants with nitrogen. And at this point, this is it from me for today. Thank you for your attention. And in case you have any questions, you can leave them in the chat. I think so, right? Or maybe we can discuss them later. I don't know in which way should we do it.
Thank you, Yaroslava, for nice presentation and interesting data. Again, I believe if there will be questions, we will see them in the chat and later can come back to them. And on this point,
Feel free to ask.
I would like to give a word to Oleg Baranovsky to continue with more practical stage of our event today to introduce us inoculant products we work with and what data we have about them. So, Oleg, the floor is
Hello,
yours.
thank you. Hello, everyone. My name is Oleg Baranovsky. I'm an agronomist at BTU. And at this point, I would like to introduce you a product for inoculation from BTU. It's a Resaline product which produced for inoculation legume crops, soy, pea, and other one. So it based on bacteria Bradyrhizobium, Rhizobium leguminosarum, Mesorhizobium, Phanerhizobium. So it's selective for each legume crops. And as you see, this product could be in two formulation, liquid and dry. So that means it could fit your application system, and also it has different shelf life, which could be better in different conditions. So some photo of our agronomist in Ukraine. When you see on this soybean was applied Rizaline, and you could see a great development of nodules on the root system of soybean. Some data about the application of Rizaline, more than 2 million hectares of
Sorry for interrupting.
yes.
Can you try to open for the full screen? Because we
So on
see
my laptop, it's open to the full one.
just maybe check if you put to show all screen and not as a presentation, because we see the first slide. If there are problems, I can switch it.
Just a second.
Mhm.
I need that.
If it doesn't work, try sharing the whole screen, not just a window, but the entire screen, and it will work in a full screen mode.
Yes. Does it work now?
Yeah, we see the second slide with our agronomist.
So great. I will continue that more than 4,000 agro companies use inoculant from BTU. The average yield increase is 0.3 ton per hectares additional yield for legume crops. And at that moment, we have more than 15 years of effective presence on the market and effective use of this product. So we have huge database about trials with our products, and we make some calculations, and here is some statistic information about application of our inoculant products. So with Rizaline, we have more than 300 trials with positive rate is 91%. Average yield increase, as I mentioned, is 0.31 ton per hectare, and average yield increase is 13%. Also, we looked how it works with different applications method, and seed treatment works a little bit better than in furrow application. So in general, it's 14.3% to 5% of yield increase. And also we compared formulation of the product, because we have it liquid and dry. And it works almost in the same rate, 14 and 13.3% of yield increase. Also, we tried a lot to combine our inoculant product with another BTU products, and in all of the cases of combination, we have positive effect. It could be with mycorrhizal product, with PK mobilizer, it's around 17%, with foliar stimulation, organic balance, also with micro elements from BTU, with name of Helprost. Also, as my colleague mentioned before, it's a new tendency of co-inoculation with N-fixer bacteria, and we tried it with azotahel product from BTU, and in average we have 22.5% of yield increase. And also with biostimulant like Humifriend, based on humic and fulvic acids, we have the biggest yield increase with together application with inoculants. And we compared how it works on different crops. Mostly it's applied on soybean, but we have also a result on chickpea, pea, bean, and lentil. So on beans we have the biggest yield increase, but on the all other crops it's also economically efficiently and have good result. Also, with application of Rizaline in BTU program, it's often recommended to apply additional products to ensure better product work. Its products Rizasafe and Liposam, which protect and save bacteria from Rizaline on the seeds. The role of Rizasafe is protect bacteria on the seed, but Liposam works like fixator on the seeds. Together, this product make protective combination for better adhesive of active substance of bacteria, and also to save bacteria longer on the seeds. And on this point, we would like to show you practical results of efficiency of different form of Rizaline application. This is trial of research institute on western part of Ukraine. Here was compared different varieties of soybean and also different fertilizer background. So in both cases, we have great yield increase with different varieties in 2 different years, and also with different fertilizer background. This was different with nitrogen application, and some small difference in yield. Here on this slide, you can scan a QR code and to watch videos of our partner about our product Rizaline in dry formulation, how they work with this product on Slovak market. And another our trial with liquid form of Rizaline, and also with combination of our PK mobilizator product ground fix. Before sowing, it was applied ground fix on pre-sowing tillage, and after that it was in furrow application of Rizaline 0.35 liters per hectares, and as a result, you see a huge increase of yield, 0.6 ton per hectares, and you can see it even on the photos. It's a huge difference of development of the plants, and you can see a lot of nodules on the root system of soybean. And another one very important topic when it comes to application, a lot of farmers want to understand how it could be applied, because some of them use chemical protectors and also want to have better result with biological products. That's why we look through our compatibility table and show you some of this part with Resalign product compatibility with chemical products. Here you can see a lot of fungicides product, some world famous brands. Also, you can see it with active substance of these products. But the main point that I want to focus your attention that you do not mix inoculants with substances containing high level of zinc, copper, and mercury. Also, fertilizer with sodium molybdate, manganese, and molybdenum, and fungicides such as prosidamidon, iprodion, metalaxyl. Also herbicide such as MCPA, 2,4-D, and Dinosep. And insecticides contain endosulfan, diametoate, ometoate, and carbofuran. And also practical results of application with chemical products. So it's not just compatible products, it also have good yield increase when it applied together with chemical protectors. You can see also these names and active substance of these products. And you see that the yield increase also good and economically efficient. And practical results with chemical protectants Lamardor and Gaucho. Control was with chemical inoculation, and trial scheme also Resalign, Resasafe as protector, and also chemical inoculation. We received almost half of ton of yield increase, and you can see how it was different in growth of the plants. Another one example with application also with chemical fungicides, Maxim XL. Also in this trial was applied protector Resasafe, and I want to put your attention on the variants 3 and 4. In this case, was applied mycorrhizal product MycoFriend, and together with Resalign, mycorrhiza performed the best. If we compare to the control, it's almost 900 kilograms of yield increase. And another very interesting trial also with our inoculant and protector chemical product. But here's another interesting thing, that on control was another inoculant. It's analog from world famous company. But you can see that on the photo, it's total difference in plant development. This photo was made by drone, and you can see it also on this photo, different development of the root system and bigger number of nodules on this root system, and also yield increase above analog products. Another one trial, also a good visual result, and not only visual, also with chemical products together, and as a result we have yield increase 0.27 ton per hectares. And one more trial with also a combination, not only chemical treatment, but also with some micro elements together. And this combination also work well in also dry condition of Odessa region. And on this point, I will give a word to my colleague to talk about seed treatment, primarily seed treatment of Rizaline.
Yeah. While my colleagues are switching slides, we have a very nice question from Lenas Dimitris Lenas. Thank you for your question and also for being with us. The question is about in field with a long history of legume cultivation, how we can assess whether indigenous bacterial populations are effective or if co-inoculation is required. So I will say my opinion, maybe Yaroslava, I will add to it. So we have such questions quite often from farmers who grow a monoculture of soybeans. And indeed, even if they don't use inoculants, they have some nitrogen fixation on the roots. But in practice, and on the example which Yaroslava showed shown in Brazil, 80% of farmers inoculate soybeans each season, and soybeans are quite widespread, and monocultures are also quite widely used of soybeans. That is why we see from practice that there is sense in inoculation of soybean seeds each season to have maximum nitrogen fixation effect. Yaroslava, what is your opinion?
I will agree with you, and what I will also like to add that potentially there is a chance that in your field there will be some efficient rhizobia which will inoculate your crop, but also chances are there are not living there and that they will not inoculate your crop, and taking this risk each year, I think it's not an economical decision, because inoculation is a pretty cheap practice, and that's why most farmers they choose not to risk. They just inoculate each year because basically don't know what's going on in your soil. So that's why they're doing it, and they see positive results. We sometimes they're also doing trials when they have on their field, they leave some part of it uninoculated and another part inoculated, and in most cases they see positive results when they actually inoculate each year, because when you inoculate your seeds with inoculant, you know that you treated your seeds with high efficient bacteria, and you can be sure that they will work. And also you have always an option to co-inoculate, for example, to apply mycorrhiza like mycofriend together with biological inoculant, and you will also increase the performance of your crop. So it's a very nice practice to implement.
Thank you, Yaroslava. So yeah, Alexander, are you ready? So please tell us about the topic of preliminary treatment of soybeans. Alexander? So while you are connecting, I will comment again that this topic is quite important. And 10-15 years when we started work with inoculants, we were recommending applying it on the day of sowing, which is efficient, but maybe not always practical for farmers. They want to do that in advance. And that is why we start to apply some protectants. As was mentioned, we have Rizosafe, and also we studied some preliminary treatments, like starting from few days and weeks and few weeks, now up to 7 weeks. So Alexander, is the microphone working?
Did you hear me well?
Yeah, we hear you and see the screen. You can continue.
Okay, good afternoon and thank you for joining. My name is Alexander Runsevich and I am a lead agronomist at B2 Biotech company. So today we focus, like my part of the presentation is focus on one of the most soybean growers treat as a routine operational detail, and I want to argue that is actually an agronomic decision with measurable consequences. When do you inoculate your seeds? So the industry default is on the day of sowing. That is the safest answer, and it's not always a practical one. Large operations need to treat seeds in advance, sometimes days in advance, sometimes weeks, because sowing windows are short and the treatment capacity is limited. So let's start with one of the foundational experiment run at Vinnytsia National Agrarian University in 2019 with the soybean variety medicine. So the design of the trial is simple: 3 treatments, uninoculated control, Rizoline with 2 liters per ton of seeds, and at this 7 days before sowing, and the same amount in dosage at before sowing at 21 days. So what we can see on the screen that let's look at the full flowering stage, which is the stage where nodulation peaks and where nitrogen fixation matters the most. So we can see that when we apply Rizoline 7 days after seeding for seed treatment, we have a robust and increased amount of nodule weight in comparison to the control site, with a huge amount, more than 300 milligrams per plant of nodule weight. And even if we postpone the seed treatment like 21 days before sowing, we still have a very positive tendency with increasing nodule weight, which is matters of the potential of nitrogen fixation. Also, if you take a look at the plant weight, we can it tells us the same story more sharply. Nodule mass at flowering is very has an increased tendency when we apply Rizoline at 7 days before sowing, or even when we apply it later, 21 days before sowing. So next one is a trial, 1 year, and one side is I would say every year we ask the same question about when we should apply Rizoline at the day of sowing. Should we postpone it for a couple of weeks, or should we even postpone it for over the months due to the operations delay or some difficulties to use it on time? So this is the same question we have tested in consecutive seasons in 2022, 23, and 24. With 3 products compared head-to-head. One is Rizoline plus Rizosave. The next variant is analog of international manufacturer. And the last variant is Ukrainian analog of bio-inoculant. So the data show us that when we apply products at the day of sowing, we have a huge potential of increasing the yield, which is give us a point every year from 2022 to 2024. And we see also the tendency when we apply, when we postpone the sowing of treated seeds for more than 3 weeks, we still have a potential to increase the yield, but with a bit lower amount. And if we, take some risk due to the operational delays and have done the treatment over 45 days before sowing, the bacteria still became alive and fix nitrogen from the atmosphere provide the minor increase in comparison to the control side. But in general, it gives us a positive picture when we between variants. If you compare Rizaline with Rizotave, if you compare with analog of international manufacturer and Ukrainian manufacturer as well. So the next, the next slide is about effect of pre-sowing treatment of soybean seeds on the total amount of number of nodule-forming bacteria, because the number is also matters, because we can count how many nodules on the roots and count how many of them are alive and doing the job. And if we compare the three variants between each other, we can see that we have a minor number of nodules at the bud formation stage of the soybean. And when it comes to the end of flowering and seed filling, it reaches its peak. And in comparison to the two analogs, like of Ukrainian and international manufacturer, we see the Rizaline shows very promising and I would say great results of the number of forming nodules on the root system of soybean. So let's go to the next slide, which is show us the mechanism. I would say count the number of nodules itself. It's I would say makes sense to do when you analyze the root system of soybean, but also if we talk about the nodules itself, we look at the mass of the nodule, because it's I would say give us an idea how productive these nodules became after the formation. And if we take a look at the three stages where we check the mass of the nodules at bud formation and flowering and seed filling, we see that Rizaline with Rizosave is an absolute leader between three variants with international analog and Ukrainian one. And at the stage of seed filling, we see the very great numbers show us that our bacteria has a big mass, and it means they are more productive for nitrogen fixation at that stage. So also we do some analysis of these 3-year trials with AI, just to see if we can extract or pick some specific information, specific tendency. So you can see that the peak nodule number between 3 years was at the third year, in 2024. And if you compare it to three products, Rizaline between competitors is showing very great result. Also, if we talk about the peak of nodule mass, Rizaline among the leaders in the trial. And also, if we touch the yield itself versus the control and versus the two analogs, we see that application of Rizaline with Rizosave is a leader in the study. So the next slide shows us when we apply products at 40, when we sow in treated seeds at 40-45 days after the treatment, after the pre-treatment, we can see the clearest competitive advantage between three products and the nodule mass. This period is very risky because from the previous slide, we have seen the tendency that the most powerful results we can reach when we apply 7 days before, or as a day of sowing, or 7 days before sowing. but at the 40-45 days, still there is a line among the leaders in the study. And if we compare the biometric advantage and translate it into the yield, so there is a line between 2023 and 2024, reach almost the highest point at the study and retain 89, 96 in nodule biomass after 40-45 days. So competitor retention was much less stable. And important to notice that stronger nodule biomass also play a role depending on the climate conditions. So the next slides will touch the theme of Frisoline in comparison to the competitive products. So we have a couple of trials, real-time data collected from 2021 at Vinnytsia region. We see that the trial is also with a simple design. Frisoline 2 liters per ton of seeds with a RisoSafe 1 liter per ton of seeds in comparison to the analog. And we see the clear numbers between the control plots and the trial plots, whereas Frisoline has more benefits in increasing to the yield up to 200 kilos per study plot. So let's go to the next slide. Okay. So here we have another real-time study where we comparison our product with analog and also when we combine it with a treatment with DMTD, is a TIRAM chemical substance. So the trial was done in Ukraine in 2018 in Poltava region on the soybean. And you can see the trial scheme where we have Frisoline plus RisoSafe, some chemical and nutrients products like TMTD and Carlet, nanovit, molybdenum, and also we have a control plots with the treatment of inoculant analog Optimize with the same chemical and nutrient products applied. And you also have you may see some very beautiful picture of where we dig and extract roots, and just to see, we wanted to see the nodules formation during the development stage of the first cotyledon leaf. We discovered that the amount of nodules potentially give us the benefits in increasing the yield, and the final results you will see in the table of the screen, where we harvest almost 600 more kilograms at trial plots in comparison to the control site. Also, when we go to the next slide, we see that another trial was done in Kirovograd region on the same crop. It's soybean. You can see that the trial scheme is same simple as it was on a few previous schemes. Control, untreated control, and Frisoline with RisoSafe, so 2 liters per ton and 1 liter per ton to protect seeds and give them more, let's say, space to work. So again, when we count the harvest, when we look at the plant, we see that we see strong benefits from the byproducts application to the soybean, especially in yield increase up to 200 kilos in comparison to the control side. Another trial that was done in Volyn region, Ukraine, in 2023. You may see on the screen very beautiful plants collected from the trial plots and control. You can see from the early stage at the June, and later when the plants developed some pods, soybean pods, so we can measure even amount of pods and amount of seeds. So the trial scheme is is also pretty simple. So we mainly look at the trial plots with Risoline and Risosave, 2 liter per ton and 1 liter per ton of seeds, and farmer technology with optimize 1.2 liters per ton of seeds. And based on the collected data from the farmer, we had a benefit from Risoline Risosave application up to 500 kilos per ton in comparison to the control area. Also, one of the most important topics is co-inoculation with Risoline and other biologicals. So co-inoculation studies is one of our interesting direction where we see how our bioinoculants work together with protectants and synergies. Especially, we have done a couple of tests with Kernel, the biggest producer of corn in Ukraine, from 2024 to 2025. We see that if we look at the control without treatment, we also have we have a couple of controls on this trial with a background. One part is treated with the biofungicide Fitobit, 1.5 liters per hectare, and another part of the variants were treated with the chemicals, Vibrance, RFC 1 liter per ton, Contador Max 0.35 liter per ton. And based on these backgrounds, both bioinoculants like Risoline and Risosave, or also some co-inoculants like Risoline, Risosave plus Humifriend, and another variant with MycoFriend, give us a strong, I would say, opinion and confidence that we not only can apply bioinoculant itself on the soybean seeds, but also we can co-inoculate it with a mycorrhizal product. We can add some nutrients with a Humifriend, humic and fulvic acids, and even apply a couple of times for Ecostern foliarly as a part of the tests. Also, if we just look at the tendency from the increased yield between variants from 2024 and 2025, we can see that co-inoculation gives us more benefits, more nutrients, and better plant development at the time of testing. Another trial where we look at the effect of seeds co-inoculation on soybean field in 2025. So it was done at the Institute of Agriculture of Western Police. So it's the western part of Ukraine in Rivne region. On low humus Chernozem, the previous crop was winter wheat, and also you can see there was some fertilization scheme with NPK. So you can see also the pH of the soil, which is also play a role in nodule formation. So between two varieties and between inoculant itself and co-inoculation with another products like AzotoHelp, HelpRost Seed, and Humifriend, we see the clear tendency. So if we can combine 2 or 3 more products together, we have more benefits for the bacteria growing, for the plant nutrition, and for the harvest yield at the end of the test. So you can see the very great numbers when we compare it into the untreated control set.
So thank you, Alexander, for your part. Again, for this part, crucial is to understand, even though different producers allow applying inoculants on the seeds for quite long period before sowing, yes, it's possible also with our formulation, but the shorter is the period, the higher is the efficacy. Also, you can see that comparing to different alternatives from international suppliers, which are also quite expensive very often, and regional suppliers, Risoline is quite competitive. And I hope that you find some ideas for yourself with possible co-inoculation of with Risoline and some other products like humic products, nitrogen fixing bacteria or mycorrhiza, indeed they improve plants' productivity quite a lot. So at that moment, I would like to ask Dmitry to share our experience of application of Rizaline also in different international trials and different crops. Dmitry, are you ready? Dmitry, we see the screen but don't hear. Try to say something.
Yeah, do you hear me well?
Yeah, we hear you well.
Do you see my screen?
We see your screen.
Okay, thank you, Dmitry. Sorry. Hello, everyone. My name is Dmitry, and I would like to ask you follow to red laser pointer, and I hope that my part will be very interesting for you because I would like to share you information how it looks in different regions, how looks work of our inoculant. As you see on this screen, you can see how it looks in Slovakia on the soybean, in Ukraine on the soybean. Please draw your attention that with our products, we can provide not only more nodules on the root system, but also our products has a good influence for create. Okay, with our products, plants can create a bigger root system, like in Ukraine with soybean. Also, few photo from Germany. This photo I will share more information a little later, but it is from the biggest one field days in Germany, which call it DLG Feldtage. As you see on the control, we have only few parts, few pieces of nodules on the root system, but with a Rizaline we had more. And also on the background, as you see, my colleagues already told you about some products which can be like synergist for the uh, inoculants, like a ground fix, and we have a better situation on the root system. Also, we have a practice to use our products on the chickpea in France, as you see. Not only a better root system. When we create a better root system, also we provide a better development of vegetative mass. This one slide, it is pictures from the DLG. As you see, control place, we have only few nodules on the root system. Where we use the Rizaline, we have more nodules. Where we use the Rizaline for the seed treatment, plus two application of our products like bioadhesive liposome and free living bacteria, nitrogen fixation bacteria for the foliar applications. We have a stronger root system, and my opinion is that this complex of products like phosphorus and potassium mobilizer ground fix for the soil treatment together with seed treatment by Rizaline can show us the best result in, let's say, in the bigger part of regions where we use a soybean Rizaline for legume crops. Also, from right side, you can see different scheme for application also show us a good result with the creation of nodules on the root system. This slide we share information that we have a cooperation with such company like ICL in Japan, and we produce products for them with nitrogen symbiotic bacteria for the legume crops. They use these products for soybean and for hail wage crop. And also, if I remember correctly, Dmitry, can you help me? We already have a cooperation with them during the last 3 years.
Good.
One more trial from not close countries. It is a trial from Burkina Faso, and we have such parameters in comparison with a control. As you see, we have seedlings, numbers of nodules per plants. For example, on the control place, as you see, was only 13 pieces. With a trial, it was with a BTU product, it was 26. For example, numbers of pots per plants. For the control, it was 51.7 per plant. With BTU product, it was 53. Also, number of seeds per plants on the control 94, on the trial around 100. And also you can see, you can calculate what different between control and trial we received. It was around 700 kilograms per hectare, increasing the yield with the BTU products. Also, very interesting trial because I think that everybody knows that in Kyrgyzstan, in Kazakhstan, we have a very difficult soil and climate condition for soybean. Why I said it, because as you see, in general, on the control place was yield was a little more than half ton per hectare. But even in these conditions, with our products, we provided additional yield plus 130 kilograms per hectare. If we calculate in a percent, it is around 24% increase in the yield. Also, this trial was bigger. As you see, the same yield of control, the same yield of our products, and they had other two scheme for soybean. As you see, moisture retention absorbent in a rate with a rate 23 kilograms per hectare provided us in comparison with the control plus 80 kilograms increase in the yield. Some analog inoculant with a rate 1 liter per ton plus growth stimulator 1 liter per ton provided us plus 110 kilograms increase in the yield. And as you already know, if you follow off my laser pointer, our products, Rizoline plus Liposam, provided us increasing the yield 130 kilograms. Near country to Kazakhstan, it is Kyrgyzstan. They use 1 hour formulation of Rizoline for beans. As you see, unfortunately we don't have numbers of yield, but you can see better development of root system on the trial plant in comparison with the control plant. A little more about chickpea. As you see, during 2 years, 2024-2025, we make the trial with different type of fertilizers. As you see, we use the phosphorus and potassium 30-30, NPK 30-30-30, and one more NPK where we have higher concentration of nitrogen until 60. It is control place. Let's say that green color, it is control place, and different result in comparison with a different type of NPK. Also the same scheme for NPK, but seeds we coated by Rizoline with the rate 2.5 liter per ton. It is like this color. Next one with the blue color, it is the same scheme with NPK, but seeds we coated by organic balance, 1.5 liter per ton plus microfriend one. It is mycorrhizal product, as my colleague already told you, 1.5 liter per ton. And last one trial, it was like all our products, like like Inoculant, Rizoline, Mycofriend, mycorrhizal product, and biostimulant, organic balance. We used it together for seed treatment. And I would like to draw your attention that this graphic show us average result during 2 years. As you see, sometimes let's say that Rizoline, when we use only Rizoline, we have a good result. But also, it depends on what conditions you have. If you have a poor soil and you need to save the money for the mineral fertilizer, I think that in this case will be better to use a complex by biological products like Mycofriend, Rizoline, and organic balance, because average increase in the yield the best for this complex of BTU products. Yeah, as you already saw this photo, it is from France. They use the liposome, or sorry, Rizoline plus Rizosafe with a rate 3 plus 1.5 liter per ton. As you see, we don't have a very high number of harvest, but if we calculate in percentages, we provide a plus 10% increase in the yield on the chickpea. About peas, also we make the trials during 2 years, and we use the Rizoline. It is as you already know, inoculant plus azote help, contained of azote help free-living nitrogen fixator bacteria, 1 liter per ton. And also for one more trial, when we for this complex added mycorrhizal product. We have two segments, like without fertilizer and with standard NPK 30-30-30. In both this situation, where we didn't have a nitrogen fertilizer, mineral fertilizer, and where we had a mineral fertilizer, in both situations we received increase in the yield with the help of BTU products. But also, I would like to draw your attention that biological products can provide you higher can have a higher efficacy where we didn't use a mineral fertilizer. And also, like Yaroslava told you 1 hour ago, that some mineral fertilizer like nitrogen can has influence for less, let's say, not so active process creation of nodules on the root system. Also, I would like to say you that with the help of this QR code, you can find articles about cover crops on our website. just to scan it and follow of this article for crops. And also our one more lab trial for effect of bioinoculants on cover crop for the germination. Sorry, please. For the germination and fresh plant biomass. As you see, let's say orange color it is control, blue color it is microfriend powder, and only one trial with a Wika. We have a green color microfriend plus Rizaline. As you see, for the germination, control place control plants was worse than plants where we use the microfriend. Again, yeah, for the Vika, Phacelia, Flex, Sudan grass, and clover, better results showed us microfriend seed treatment before sowing. And as you see, this green color, it was microfriend mycorrhizal product plus inoculant. The same situation with biomass, with action and yeah, action for the bigger amount of biomass. Orange color, it is mass of the underground part, control place. Sorry, control underground part and above ground part. For the microfriend, it was higher, but tendency is the same like on the control. And also, as you see, complex line microfriend and resalign show us the same tendency, but in this case, underground part was show us the best result. And also it is one more trial, and I would like to ask my colleague Yaroslava to help me with the best explanation what we did with that. Yaroslava?
Yes, so in a few words about this experiment. So we used tool to measure fluorescence of chlorophyll as a marker of photosynthesis activity. So just in a few words, what we measure here. So we know all chlorophyll that is in chloroplasts, which is very important for photosynthesis. And when light is perceived by chlorophyll, there are three potential fates what may happen to chlorophyll. So chlorophyll may transfer this energy for photosynthetic reaction. It may dissipate it as heat, and it can also use this energy for fluorescence. And the higher is the fluorescence, it is so high fluorescence is basically marker of lower photosynthetic activity, because we see that more energy is used not for photosynthesis but for fluorescence. And we have a tool which is called FluoraTest, which allows to measure chlorophyll fluorescence in darkness adapted leaves. So we cover leaves for some time, and after that we turn light on in this tool and measure fluorescence in leaf and see some parameters. So at this point, this is starting fluorescence. When photosynthesis is not going on, then we see a peak fluorescence, and after that we see a plateau and some stable condition. And the higher is the maximum fluorescence, it's actually the better. And because it means photosynthetic processes have a good capacity of productivity. And as you can see, the variant which was treated with mycophren together with inoculant rizoline had demonstrated us the best results in it. So you can also see this parameter F maximal, so it's maximal fluorescence after light is on, and variable, so variable is when we so it's maximal minus 0.1, and the higher is this ratio, it means a better productivity, and we see that two experiment variants were better than control, and the one where we used mycophren with rizoline was the best. You can actually see this parameter when you are reading some reports of field testing. It's a pretty important parameter, so maybe some of you are already familiar with that, and this one is the most interesting for us. So something like this.
Thank you, Yaroslava.
you.
And on that, again, I would just show something as a kind of summary. Thank you, Dmitro, as well. Moment. So, as you see, we have a lot of examples and experiments and data. And so sorry for taking so long for this webinar, but really we want to share with you maximum of our experience. And we already had some requests about recording, so it will be available online on our YouTube channel. So our idea is to gather maximum of our data and share it, make it available for our customers and our partners. Also share the presentations. But also we try to gather the most relevant and most comprehensive data in some simple marketing materials. So you can find with these QR codes a brochure, simple brochure, and but very illustrative for our experience. So we show here examples of efficacy in different countries and also examples in comparison to competitive products and preliminary seed treatment. But also, I again want to show you please use our YouTube channel as well as our website, because here you can find most of our previous webinars on different topics and also presentations from our different international events. So on that, I would like to thank you for for being with us today. During the most part of our webinar, we have around 40 participants, and we were very glad to have all of you. By the way, if you are still online, you can show yourself, open the video for us. But again, I counted around 40 participants today from 14 countries, from different regions, from Europe, from Africa, from Asia, namely like Bulgaria, Egypt, Morocco, Germany, Ukraine, Greece, Austria, Portugal, Kenya, Cyprus, Turkey, Slovakia, Jordan, and Algeria. If I missed somebody, sorry. You can write about yourself also in the chat. But again, thank you very much for participants. Soon, recording of webinar will be available on our YouTube channel. If you have any questions about Rizoline, inoculants, or any other biologicals, you are welcome to write us on email or in any messengers. We are here for you. And again, I believe this we will continue this practice, and so follow our social networks, news, and we will invite you to our new sessions. So thank you again for participation, and see you soon on our next events.
Thank you. Bye.